Learn about the newest digital pathology trends in science and industry, meet the most interesting people in the niche, and gain insights relevant to your own projects. Here is where pathology meets computer science. You are listening to the Digital Pathology Podcast with your host, Dr. Alexandra Zhurov.
Hi, Tom. Welcome to the Digital Pathology Podcast. How are you today?
Good. Hi, Alexandra. How are you?
Great. Thank you. Thanks for being my guest. And tell our listeners a little bit about your background and a little bit about where you work.
So I have a PhD from genetics and cell biology. My thesis was on cell cycle and transcription in a variety of model organisms, really. And then I did a postdoc at Dana-Farber Cancer Institute here in Boston under mentorship of Dr. Miles Brown. And that was on genomics and of breast cancer, genomics and epigenomics of breast cancer and prostate cancer. So that was really my inroads into big data and more translational research. And while I was doing my postdoc, I was also doing some pitch competitions and other form of investor relations and business development courses that I found kind of stimulating and interesting. And through that work, I got into contact and doing some consultation work on digital pathology and indirectly got into touch with Iforia and they liked some of the stuff that I was doing and a couple of years later, a year later, and they recruited me to start working together with them to bring about this the AI transformation of digital pathology.
Mm-hmm. So what is Iforia? What do you do at Iforia and what do you offer?
So Iforia, the name comes from AI for image analysis, Iforia. Mm-hmm. And we are a platform and solutions company that provide intelligent image analysis solutions for digital pathology as well as really any other image centered problem. So while we are focusing on the medical imaging field and medical questions and medical research, especially in the US, there is our platform is completely agnostic. So we have done projects also one-off basis in different other verticals, aquaculture and industry, other industrial processes. So it really is centering on automating image analysis, but obviously we are interested in focusing on digital pathology and other forms of medical imaging.
Mm-hmm. Okay, so digital pathology, image analysis for digital pathology. Image analysis for digital pathology is kind of booming nowadays. And I wanted to talk to you about the validation of the image analysis solutions, and especially artificial intelligence-based image analysis solution. What is validation and what types of validations are there?
I think that in any assay that you develop or any assay that you start using, finding the boundaries of the usability or the precision and recall of that assay is really the most important aspect. And it doesn't really matter if it's artificial intelligence or if it's images or if some other form of analyte. This part of the process is always, important and kind of the same. Mm-hmm. So we have first of all we have validation, like conceptual validation that can the platform or can the technology do what you want it to do. So that's a trust building exercise, right? Mm-hmm. And then we have when the assay is ready or the device is ready, and then we have the analytical validation. And an analytical validation really means that you can precisely quantify the accuracy of the method for measuring the different, let's call them analytes. So in the case of digital pathology and artificial intelligence, if you design a tumor grading algorithm and you have a ground truth, so you have some knowledge that you can apply to images to quantify, to say where are different tumor grades, what are their size, and which lesions belong to which category. Then you compare your human knowledge and your human annotations to that of the AI. And for each of the things that you measure and quantify using the artificial intelligence, you have one output. And so for grade 1, you would have a certain amount of precision, and for grade 2, you would have another class of precision or another precision, and so on and so forth. And so then this constitutes the analytical validation, and you can say that overall the precision and recall or F1 score or area under curve or anything else that you want to use as a measure comes to a certain threshold for that particular validation set.
So this is against ground truth. So let's say human annotations, if we would.
Yeah, so precisely.
The pathology example.
Yeah. And so ground truth is a concept that gets thrown around a lot in artificial intelligence, and that's really only kind of the human definition or our usual gold standard definition of what things are. And ground truth is what we are using to learn to teach the AI. And then we compare again the AI towards ground truth, and then when we are doing the analytical validation, a lot of times and a lot of AI publications out there are weak because they are not being validated according to the best standards. Quite often, when you train AI models, what you see is that you do a holdout data set and you do a training data set. So the training data set becomes what generates the first AI model. So you provide labels in some form from the training data set, you generate a model. And then in machine learning and AI, quite often validation is a stage that they define as, so the term is used when you are using some part of a holdout data set to improve the model. So you're adjusting the weights or some hyperparameters in order to fit the model to work better on this other data set. And this is different from what most biologists or people in the medical profession call validation, where you try to validate a method. So these, sometimes it's hard to keep these concepts apart. So for the purpose of this discussion, I think that when we talk about validation, we talk about validating the actual assay as a biologist or a scientist would understand it. And when we talk about the other form of validation where we are improving the AI model, then let's just talk about improving the AI model or adjusting the AI model or tuning the AI model.
So what would be assay validation in this image analysis context?
I think that assay validation is the analytical validation. So when you have your trained, tuned, adjusted AI model, you take a external data set that has not been part of any stage of training or tuning, and then you do the analytical validation for the metrics, wherever anything that you measure in the AI model, and let me see, I need to try to formulate this now. So when your training is ready and you believe in your model, you have tested it to some extent already on some external data set, and you know that it generalizes quite well. At that point, you take a completely external data set that so far has been in no part of this development process, and you compare directly on a pixel level what is the agreement between humans for their diagnosis or their scoring of different areas of the image, and compare that to what the AI is outputting. And then that percentage agreement becomes the direct score. And so then you can do this confusion map where you have true positives, false positives, false negatives, and true negatives. And based on those numbers, you can derive precision and accuracy, precision and recall, and from that then the F1 score. And of course, the way you can tune these assays depending on what your need is. So you always need to think about the so-called intended use when you are first designing the model, but especially when you're validating the model. So you need to design your validation strategy so that it actually matches what you are going to use the AI model for. And so if you, as an example, if you are looking for a screening tool to the needle in the haystack, so you want to go through 1,000 cases and you want to find, if you want to screen those 1,000 cases, to narrow down to, let's say, 50 cases that might or might have what you are looking for. Then you're looking for high sensitivity, but it does not need to be very accurate, because if there are some false positive, that's okay. But you don't want to miss something and lose out, because it's kind of a triaging method. And so, but then again, if you're using it for diagnosis, and the AI model would direct some really, really monumentous or important decision for that case, then you want to have very high precision, and the sensitivity might be something that you are okay with sacrificing on. And so a lot of development becomes a balancing act between accuracy and precision, because different intended use requires a focus on different aspects of this. So I think that it's very important to not be intimidated by AI models and think that the validation has to be in some way different. I think that any assay really gets validated within the same framework. When it comes to AI models, you need to be cognizant of how an AI model is being generated, so that you actually measure then the right thing, or that you validate on the right set of data. It's quite often when you are seeing new products or new technologies, they are initially presented in a best case scenario, obviously. And so while I was doing my postdoc and my PhD, there's always new products, and there's always new leaps in instrumentation that can accelerate your research and make it faster, cheaper, better, even enable things that you never saw before. I think the emergence of next generation sequencing was a boon. I started my postdoc around those times when that started really to take off, and so when you get presented with the new technology, there's a lot of hopes and dreams, and obviously also promises, and being able to validate those on the things that you actually are going to use the instrument or the technology for is very important. Because if you only look at the validation from a best case scenario, then you necessarily won't know what the validity of the method is for your particular intended use. So either you have to use it in the exactly same environment as it was tested for, or you need to validate it for your particular environment.
Mm-hmm.
And I think that this is very true in the AR world, where there's a lot of applications out there that are not properly validating their methods, and when they are replicated, or when somebody tries to externally validate these methods, they don't necessarily perform as well.
Okay.
Yeah.
I think you mentioned two very important concepts: the intended use, that you always have to be focused on your intended use, and that it's like any other test or any other assay, and the validation is as any other validation. So there's no need for reinventing the wheel. There are principles for test validations, like you mentioned, that favor either precision or sensitivity depending on the intended use. And this is these are the guidelines that we can use for AI validation as well.
Yeah, absolutely. I think that there's really no magic, and I think that there's just
that's a good one as well. It's no magic, although I think in many minds it seems like magic, but no, it's just another method.
It's still science, and it's still a tool. It's a very powerful tool, and I think that where we sometimes do get lost is assuming that the AI is doing things that we did not train it to do. So let's say that we have an AI for some pathology, let's say liver pathology, NASH or NAFLD. So then if we say we have an AI for NASH, then what does that actually mean? Do we have can it quantitate certain measures of NASH? Does it provide a digital SAF score? Or can it have magically, create a diagnosis of NASH without anything else? And so there's three possibilities here. One is that it's some independent scores, maybe not a full like battery of SAF criteria, or then it's kind of a correlate of what pathologists are doing today, which would be the SAF score, or then it's something that's kind of beyond human capabilities, which is just looking at the image and leaving everything as in a black box, and then saying that the diagnosis is this. Mm-hmm. And so I think that most AI today is somewhere in that middle ground, where it provides a very exact quantitative score, maybe sees things that are difficult to discern for a human, especially it's difficult to discern precisely and reproducibly. And then of course we have in some cases where we already today where there are things that are not really seen by humans, but by some means can be detected by an AI. And so individual scores, the lowest level, that's kind of easy to validate because it's just one score and it's a precise measure, and the intended use is quite clear. Then we come to the more complex cases where it's a combination of score or digital SAF, where you need to first have a validity of all the individual measures, and then also a validity that the measures put together actually correlates or mimics what would happen in the clinic or what would happen in the research setting. And if there are differences, of course, there is really no reason to believe that the AI would be more wrong than a single human. Rather, what you want to do is compare to multiple different humans so that you see what is the dispersion of opinions. And then, of course, then in the black box, kind of like beyond human, just get one value for one sample, or that would be the diagnosis or the predicted outcome. Then that requires a higher level of validation again, because you're measuring so many intangible things that is hard to quantitate, and there's also a lot of more chances for confounders or biases that you have not taken into account during the training period. So that's why currently I think that the safest best use of AI is providing this beyond human capability assaying of things that we know are there, and we know that they have biological meaning, and we are kind of beginning to scratch on the surface of these beyond human cases. There's one good example that I was at a conference last week at TriCon in San Francisco, and I was introducing Eric Topol, who gave the keynote, and he was referencing to this example where just from images of the retina of the human eye, there's an AI that, with some degree of precision, can guess the sex of the person from just based on the retina image. Mm-hmm. Nobody knows why or what are the features that the AI is looking at, but it seems to be, within some boundaries of confidence to be accurate.
Mm-hmm. So how do you validate this? It's something where you don't have a ground truth to train for. And like you said, we're kind of touching the beyond human capabilities, the information that is not perceivable by human eye, but is in the pixels. We talked about the analytical validation, and I assume that would be validating against outcome or against something that is independent, like another method that detects the same things.
Yeah, so I mean, in this kind of artificial example of basically determining the sex from a retinal image, you should test according to outcome prediction, right? Mm-hmm. So there is first you can test on the training set, you can test on the tuning set, or a holdout set that kind of is part of the training set, but that kind of extracted from the training set but has never been part of training. But those validations tend to overestimate the precision and accuracy of any given model, mm-hmm. because it's kind of similar, and there might be some statistical noise patterns in that data that correlates highly with your binary outcome. So in this mm-hmm. case, male or female.
Yeah, I heard about an example.
Yeah, go ahead.
Where a pathologist would train for something on images that were manually annotated with a black dot. And yep. This black dot was the feature that the model learned, and it was very precise, but it had nothing to do with what was in the images.
Yeah, no, this is a very, very common occurrence, and this is why very loosely supervised training is fraught with a lot of mistakes. In medical literature, there's tons of these mistakes out there. There's some spurious signal that can be either a confounding signal like the black dot, maybe there's two, like maybe there's two different kinds of instruments from two completely different populations of patients or cases, where one instrument has a bias towards less severe cases, and the other instrument has a bias towards more severe cases, and then the artificial intelligence will learn that the instrument information that is on the image that is the strongest signal, because the signal then is very, very precise, because usually it's a serial number or something else, mm-hmm. Or model number that is very, very exact, and artificial intelligence looks for of labels, and so this is why this kind of loosely supervised training is sometimes a little bit risky. It's better to do some level of supervision so that at least you are excluding large parts of the noise. And I think that especially in digital pathology, there is a very high risk of training to noise, because if you use whole slide images and train only according to outcome per image, there's a lot of information. These images are hundreds, can be 100,000 pixels times 100,000 pixels. Some of the largest images that we deal with is almost 300,000 times 300,000 pixels. And this is an enormous amount of data. These are like 5,000 like modern mobile phone images. So there's like 5,050 megapixel resolution images, or something in the ballpark of 15 hours of HD movie. So there's an immense amount of data in there, and only a small fraction of those pixels are actually meaningful for the disease, and everything else is noise. And so I think that my background in genomics served me particularly well also in my current role, is that we were dealing with so much information and so much data that you always know that whatever analysis you do, you know that you have a certain amount of false positives. Even if your method is extremely precise, you still will have it on a whole genome sequence level. You'll have thousands of false positives. So how do you find within that mass of false positive or noise, do you find the actual signal and separate out the signal that might not be actually the strongest one from a statistical point of view or from a technology point of view? And the very same is true here in the image analysis field with the large images, where the signal is a very small fraction of the actual data that is there. So the signal to noise ratio is very, very skewed. And that is why the Euphoria strategy, well, we predominantly do tightly supervised AI models, in addition to less supervised methods. And so a lot of the success that we've had so far are precisely from these supervised methods, because you are already by your human ground truth, you are honing in on the very the details that you know are meaningful for whatever you are studying.
I think this is also touching on kind of a fear that, is AI going to take our jobs? Like you say, it is so prone to bias when it's not supervised that it's very unlikely to use it without a human expert that can sort through the information and decide what's meaningful or not.
Yeah, I guess that, there's a couple of different things. I think that first of all, I don't think that any particular very highly technical profession is in immediate danger of being replaced by AI. Especially in a highly regulated field such as medicine or medical research. I do think that anybody using the most advanced and most modern and the best tools, including AI, will replace the people who are resistant to using the best tools. So getting on the AI train is probably a good idea, because why would you not be as efficient and as good at doing research or providing healthcare as you can? But in the long term, I do think that a lot of the things that you and me do today will be replaced by AI. And then industrial revolutions have happened, throughout history, where we invented the wheel and all of a sudden people didn't really want to carry things anymore, right? We had a huge industry in shipping where stevedores were offloading and loading ships. But today, most of that activity is automated. It's robotics, it's containers, it's cranes. It's like all of that is just being managed by a very complex automated system. And I think that AI is doing the same thing with a lot of different industries, and I do think in research the same. It just means that we don't have a lot of longshoremen and stevedores that are walking around unemployed and wondering what they're going to do next. We always, we as society, we kind of go through these industrial revolutionary changes, and things change, and we change with them.
And
like
change,
my parents' jobs are not my generation's jobs, and the following generation will not do the same thing that you and me are doing today. Some of the aspects that we are doing, they will continue doing, but some of the things they will come back and see that, oh, thank God I did not have to count those nuclei manually and get,
I think it's a very, very important perspective too.
Yeah. So I do hope that AI will replace, most of what we do. I think that, from an ethics standpoint, the only thing that I would like for AI not to replace is the arts and, sports and things like this. I think that, these are things that we should allow humans to have kind of, unique rights, things that are fun. Let's do things that are fun and fulfilling, and let's have automated system do things that are menial, boring, tiring, causing depressions. I think that it's not about if AI is going to replace us. It's like, let's think about what we want AI to replace and work towards that goal. It's a little bit of a segue from validation, but I do think that AI has a really important role, but only through validation. Because if we don't validate these methods, we're gonna produce a lot of garbage.
Exactly. What are the dangers of not validating? You say producing garbage, unreliable data.
Yes,
what else?
Yeah. I think in research there is a huge risk for going down the wrong track, and it obviously happens a lot already without AI. So you do a method, you get a some preliminary result, and you validate it, but you are maybe a little bit too optimistic, or you use the wrong validation method. You're under pressure, you need to deliver, and things get a little bit their own life. So then you continue down a path of research that is based on the wrong premises. And with AI, it would be kind of the same. You have an AI model and you assume and it's measuring something, but actually that was not the intended use. So you're using the device for the wrong use, not for its intended use. But you still assume that it's accurately measuring what you want measured. And so if you are doing this, then you're gonna make unsound assumptions for the data that you receive. And so again, it's true that if you have a method and the intended use is A, but you validate it for B, then it can only be used for B. But if you believe and everybody else believes that it's validated for intended use A, and go ahead and use that, you're gonna get problems. Mm-hmm. And the further A and B, the intended uses, are apart, the bigger the likelihood of errors are, and of course the bigger likelihood of you noticing that something is amiss is as well. So I think that there's for validation there's multiple different things. First is to have just the kind of trust validation in the beginning that this can work. Okay, so then you start developing it, then you validate for intended use on a completely external data set. But then even after that, and you do it correctly, so your intended use is A, you validate it for A, and you continue to use it for A. Then as you continue working, you still need to have some level of quality assurance in place, where you either take a couple of cases, % of cases or 5% of cases, to actually verify that this automated method is continuously providing the correct result. And after a while, you will find situations where the precision is lower than what validation would suggest. And at that point, it's up to the user or, the instrument provider, whatever the arrangement is, to actually analyze the cases where precision and recall is lower than expected and figure out wherein the problem lies and how that problem can be mitigated. And so I think that this, there is the AI model has, or AI technology have a number of different solutions to actually use this data that is not performing to standard in order to improve the model and continuously become better. And at some point, you're going to have an AI model that will actually perform above the needed threshold in any case. But so before you get there, you need to have a quality assurance step in place, because otherwise, again, it's
so
this.
Quality assurance is going to be something that is implemented in the use of the model, the device, the system, after you have validated and you have proven that it's actually doing its job.
Yes.
So it's like a continuous process to monitor that it's working according to its intended use.
Yeah. And of course, there's I think that there's today with any and all instrument, almost throughout any industry, there's always quality assurance steps in place. It doesn't matter if it's AI or non-AI, because there are so many things that go into the result that comes out of an instrument. What are the pre-analytical variables? Did they change? Is the material different? So did the staining change? Did the autostainer, go down and get repaired, and then all of a sudden it provides a slightly different, stain intensity or something like that? These kind of systematical pre-analytical variables, if they change, and of course in the real world they quite often do. So then there needs to be a quality assurance that can capture changes in the outcome, regardless of if it comes from pre-analytical or pre-instrument variables, or if it comes from the actual instrument itself. The instruments can break. It's not that likely a case in AI, but in like other instruments, it's more true. But I think that there are so many things that can go wrong in a complex world that you really need to have a robust quality assurance in place, because otherwise things might change and you don't know it, and then you are you're depending on results that are dependent on an assumption that is no longer true, and then of course this can lead to all kinds of downstream effects that you don't want to have. And this is of course true in research, and it's true in industry, it's true in healthcare delivery, it's true anywhere.
So I know the Aphoria platform has some analytical validation tools that the users can use. What are these tools that the Aphoria users can benefit from when they're designing their AI algorithms?
Yeah, so at the end of last year, we released a feature specifically intended to help users validate their system at the object or pixel level. So before we have done validation according to human scorers, quite robustly, but it's a relatively tedious process. And by automating this, we hope to facilitate more robust analytical validation of AI models. So in practical terms, and short practical terms, what you do is, after an AI model is done and you basically freeze it into a stable release. Let's call it version 1. So when you have that decided, then the developers and the end users pull out the holdout data set that has never been even close to the AI model design. And then you decide on a suitable amount of material, so how many areas in how many slides and how big these areas are. And the limitation to how much gets validated is really not dependent on the AI, because obviously the AI can analyze everything and it can analyze, millions of slides if need be. It's just a matter of computational power. It's more comes down to the capacity of a human to actually provide the ground truth, or for the human to actually accurately score. Mm-hmm. And so the less areas or the less amount you actually score by a human, the better, the more precision the human has. But the less the validity is smaller, because you are testing on a smaller subset. So it's good to push a little bit, so that do the maximum amount that your collaborators can do, in an afternoon, because then after that, if they takes multiple days to do it, then they get bored, and maybe you can't get stakeholders to sign on to actually do it. So there's a little bit of a balance between needs and what you actually can achieve by human means. And so after you have decided the areas and the amount that gets validated, and you assign those regions, and you can do this very quickly and easily straight on the platform. You just enter the email address of your collaborators or your validators into the platform, and an invite will go out to that email address. And just by clicking on a link, the user will be instructed to sign in, and the user will then come into a restricted area for that user only, and where that user will only see the entire slide in the case of this digital pathology images, and they will see which are the regions and a list of regions where he or she is supposed to provide a score. And so then by clicking through the list of these regions, you'll automatically get taken to the regions. You provide the score for all the different AI model layers and classes, all the different analytes, essentially. Mm-hmm. And when then you can monitor, you as a study director, you'll see everybody's scores, but the individual validators are blinded. So they only see basically their own score that they have given, and they see the slide, but they don't see the training, they don't see other validators' scores, so they're completely isolated opinions.
And this is all incorporated in the platform.
Yeah, this is all happening natively on the platform. Mm-hmm. And so then you as a study director, then you press one button that says run validation, and that will initiate an analysis where the AI also analyzes the same regions and compares itself at the pixel level to the scorers. So then you get a precise percent pixel agreement between AI model and the different validators. And
so
I think
how many validators can you invite, or what is recommended?
Excellent question. I would say don't do less than 3. Uh-huh. I think that the absolute minimum is 2. Never mm-hmm. only validate against one opinion, because then AI and human will have some level of disagreement and you cannot quantitate it. Okay. But if you have 3 validators, then you have a good idea of what is the variation between humans. So then you get an objective score of what is the accuracy of the human method. And then you can see if the AI model is outside, is away from all the 3 in agreement, or is it agreeing with 2 persons but disagreeing with 1 person. Mm-hmm. You can actually plot that data and see if the the humans cluster away from the AI, or if they're all clustered together.
You can also plot the validators against each other, right?
Yes, exactly.
To see if you have somebody who's an outlier. And
yeah. So then you can find who is in high disagreement, and then of course after you find that somebody is in high disagreement, then you can go and have a look at the validation regions, look at everybody's score, and try to find out why validator number 3 was an outlier. Is he more correct than everybody else? Maybe he's the best. He's, the person that has created the scoring system, as an example.
Okay.
And everybody
or the other way around.
Or the other way around.
Was lazy and didn't want to do a nice validation.
Yeah, maybe that person had a bad day. These things can happen, and that's why it's really important never to go. You never go to sea with one compass, and you don't go to sea with two compasses. If you have one compass, if it's broken, you get lost. If you have two compasses, one compass is broken and one is correct, you'll never know which one is the correct one, right? But if you have 3, the likelihood of 2 compasses breaking in the same way is unlikely. So then you'll have either 3 disagreeing, and that you need to, look at stars or something, or then you have 2 that are agreeing, and then it's very likely that the 2 that agree are actually following ground truth, and they are pointing towards true north.
So last question, what are the resources that you would recommend the listeners to use when they are designing their validation? Are there any publications that are your favorite, any courses, anything that I can later put in the show notes so that people can go and use these resources?
Yeah, so I think that the best resource that any scientist or pathologist have for doing validation is actually their own robust education and experience, because like we discussed, validating AI models is just as validating everything else. But there are some reading out there, and it's such a very dynamic and fastly developing field that mostly books are kind of out of date by the time they are published. But so there's a series of online resources that I'd recommend for like general AI and also for validation. There are some courses on Class Central, edX courses. So you have some. There's University of San Diego, there's Stanford, there's MIT. I think that a lot of what we do is mostly kind of biostatistics ideas or concepts. So I think that some of that literature is really good as well. There's Coursera. Coursera has 160 something courses on AI models for different domains, and some of them have validation. I guess that because validation is kind of such a similar concept throughout different technologies, it's there's not a lot of this is how you validate practical protocols out there. There are baked into a lot of different, but so I can recommend so from Coursera, Andrew Ng, convolutional neural networks. There's AI for everyone by the same guy. We have Rav Ahuja from IBM has AI foundations for everyone, also on Coursera. So there's a lot of different online resources and courses. And then
I will put some of them in the show notes so that.
And then I'd follow the kind of medical literature. So there's a Lancet review that it's a systematic review that is very interesting. There's some FDA guidances on it. So this is something that really highly recommended reading. And we have one thing that I would like to promote. I was originally born in Finland, and there's a Finnish government initiative to basically teach the entire population the basics of AI. And this
I heard about this course. I heard that you can just sign up and it's for free for whoever wants to learn.
It's an excellent course. There's different languages. Let's see now. There's 1, 2, 3, 4, 5. There's 6 different languages currently that the course is provided in. There are Nordic languages and English so far. And then, so there's more than 350,000 people have already taken this course. And it's being kind of broadcasted and promoted at the European level now as well. This is a good kind of, it's a kind of easy, fast way to get into the basics of AI. And I think it is really important to learn the different terms and of the technology, so that you can be informed about what are the different aspects of AI that people are talking about. Because then when you go into designing the validation, it's really important that you don't go in with wrong assumptions about what are the things that actually should be validated. And understanding a little bit on how AI models is derived will help you to avoid some of the overfitting or validating to the wrong intended use pitfalls that do happen. But it's really with any assay, you just need to have a very clear idea of what the system was designed for, what you are intended to use it for, and take it from there. But so we'll post a list of these potential reading materials. Correct. There's a ton out there, and again, more is coming out all the time. So I think that Google and YouTube is really your friend. There's really no single textbook that can help everyone or everything in this field yet. Probably at some point there's going to start being that body of literature as well. And then furthermore, I think that one of the things that I have found most helpful for my own work in this field is actually just reading. There's a book. It's called Practical Statistics for Data Scientists, and I have another one that is Introduction to Statistical Learning. These are books the books that I use are heavily geared towards the programming language R, which is very useful. It's like kind of, you don't really need to know much about computers in order to quite quickly be able to utilize these tools. And it's really, really helpful because you can do statistical tests easily. You can do data visualization quite easily with these, and that really also helps with understanding the data patterns and potential biases and in your data and in your training model. So I'll provide a list of my favorite.
And I'm gonna post it in the show notes. Tom, thank you so much for this discussion. I bet we could spend another hour going more into detail of all the aspects. Just tell the listeners, where can they find more about Aphoria online?
So you can go have a look at aphoria.com, so AI for IA.com. That will take you to the main website, and there's a lot of different use cases, some knowledge documents, concepts of AI that you can read about, and there's also links on how to get in touch with an actual human or me on that website, and we'll be happy to be part of your validation, discussion and planning.
Thank you so much. Thank you for being my guest, and I wish you a great day.
Thank you, Alexandra. Thank you for a great discussion, and have a great rest of your day.
Bye.
Bye.
Thank you for listening. For more great digital pathology resources, visit the Digital Pathology Consulting website and subscribe to our newsletter on digitalpathologyconsulting.com. After subscribing, you will get access to the free annotation guidelines, which will help you annotate slides consistently in all your digital pathology projects. Talk to you in the next episode.